Hi class, welcome to our second series of lectures for week 2, Cells and Structures, an Introduction to Neuroanatomy. In this first lecture, we're going to examine the cellular components of the nervous system. Our learning objectives are to identify the 4 principal divisions of the neuron and their main functions, and describe the role of the second prominent cell type in the nervous system, the glial cells. Scientists knew very little about neurons in the 1870s, where 3 inventions in the late 19th century aided investigators to gain a better understanding of the nervous system. First, they needed better microscopes that could magnify without distortion. Second, they needed improved histological techniques to see cell bodies and their processes stand out from their backgrounds. And third, they needed intriguing investigators with the willingness to look at the samples with an open mind. Now, the first improvements in microscopy occurred in the middle of the 1800s with the development of the condenser. And what we're looking at here is a drawing in one of the first textbooks to visualize or draw out some of the primary cells of the nervous system, the neuron. And these were produced by the French physician Louis-Antoine Ranvier. So he was using some staining methods at that time that could show you some individual cell types. However, the staining techniques were lacking in the ability to show you some detail. I kind of want you to imagine that you're looking down at brain tissue. Brain tissue is really like fatty, and if you looked at it under a microscope, there would be a couple of things going on. One, you wouldn't be able to see much of anything at all. It's kind of this opaque substance. Two, it's pretty squishy, so it's hard to cut. There were techniques developed to both preserve brain tissue, to make it more amenable to cutting by formaldehyde, for example, makes the tissue harder. Improvements in microscopy, as I just mentioned, and the other thing was the development of new staining techniques that could give us a better visualization of individual cells. That new staining technique occurred by the work of Camille Golgi, who had developed a silver nitrate stain. Now, this was a very interesting stain because when brain tissue is exposed to silver nitrate, it stains in very vivid black, only a few cells that it comes into contact with, leaving this kind of orange background. There's more than 2 or 3 cells in this picture. There's probably hundreds, if not thousands of cells, but we're only visualizing a couple of cells because of this random staining technique called the silver nitrate. But what it does is it allows you to see in very vivid detail the individual cells of the nervous system. What we're looking at here are neurons stained with the Golgi technique. A second prominent researcher at that time was using Golgi's silver nitrate stain and examining these neurons in a lot more detail. Here we're looking at a close-up of a Golgi stain, and what you can see are not only are these kind of individual blobs here, which would be the neuron cell bodies, but there are these various processes coming out. And then even on those processes, there are these little hair-like structures. Those are dendrites and dendritic spines. We'll explain in a little bit more detail what those structures are. But with the Golgi stain and good magnification, you can really look and examine at neurons in pretty good detail. The other researcher at that time was using Golgi stain to examine this tissue under the microscope. Here are some more drawings by Golgi himself. You can see that there are these structures that are kind of going around. One thing, this is looking in the cerebellum, so we can now look at this and say, oh, those are these Purkinje cells in the cerebellum. But notice that their processes are all kind of interconnected and intertwined. In fact, with Golgi's work, he thought that each of these processes coming off of the individual cells were interconnected and forming what he called a nerve net. Finally, the other scientist that I wanted to address was Ramon Santiago y Cajal, who was a Spanish neuroscientist, also quite a talented artist. These are pictures that he drew, but he used the Golgi technique on younger animal tissue and could draw out in exquisite detail some of these structures. We can look at this structure today and say this is the hippocampus. We can see the dentate gyrus in different areas of the hippocampus, CA1, 2, and 3 in the entorhinal cortex. Cajal was very fascinated by what he was seeing under the microscope, that he inferred a line of communication. He thought that some areas of the cells that he was seeing, these are Purkinje cells in the cerebellum, would receive information, and then a single process coming off of that would send information. And although they were looking at similar images, Cajal thought that these individual dendritic spines must be places that come into contact with that single process, which we now call the axon, forming a connection. So Cajal believed that the cells were not interconnected, but made contacts with the space in between. Sherrington would later call this space the synapse. Cajal and Golgi contributed a lot to our understanding of the cells. Cajal himself came up with what he called the neuron doctrine, that the nervous system is made up of discrete individual cells. The two main cell types are the neurons, which were visualized there, and the supporting cells called glia. We'll be talking about those in more detail. Let's look at kind of a modern representation or depiction of the neuron and see what different components we can see within the structure of the neuron. So the neuron is an individual cell. It's like any other cell in your body. There's a nucleus that has genetic material. It has a membrane allowing the different components of the cell to be encapsulated in a similar area. It produces energy and mitochondria, ribosomes to produce protein, all of that wonderful stuff. But neurons are specialized cells, and they're the single functional unit of the nervous system. What we can see here is that the cell body has these processes coming off at the top. These are called dendrites. Dendrite comes from the Latin word branch. If you ever go to, let's say, the park in the winter time and you look up at an oak tree, that kind of reminisces of what these dendrites might be. There are these branches that come off. And then off of the cell body, there's a single long projection, which we call the axon. And then at the end of the axon may split into multiple areas called axon collaterals, but at the very end there are these axon terminals. Now the axon terminals are going to form connections with dendritic spines on the next neuron, kind of forming a connection. Now notice there is a pattern of flow of information. Information is received in the dendrites, so it's the input. Then that information is then processed in the cell body, called integration. That information then is sent down along the axon, the conduction zone, and then the output is the axon terminals themselves. So we can kind of imagine that each individual neuron is an information processing unit within a larger structure of the brain. Now, you can identify neurons based on both structure, how they look, and functions that they have. So let's look at some examples of these. There are three general neuron types within the nervous system. There are motor neurons. Now, motor neurons are the main output of the nervous system. Motor neurons will have cell bodies within either the brain or in the spinal cord, and they will send their axons out to innervate or come into contact with muscle fibers out in the periphery. So motor neurons are the main output. Sensory neurons are the main input to the nervous system. They detect sensory information in the periphery and send it into the nervous system. So if you have an output, motor neurons, and an input, sensory neurons, everything else in between we just collectively call interneurons. And interneurons will make up the bulk of what we see in the brain and even the spinal cord. So let's look at some of the functions. The motor neurons, again, are the major output. They start in the spinal cord and they send their axon out into the periphery to make contact with muscle fibers. Every time you move an individual muscle, it results because of the firing of a motor neuron. And motor neurons have the stereotypical shape where here's the cell body, the dendrites, they send their information along the axon, which then makes contact with muscle fibers. The other cell type that we described is a sensory neuron. Now, sensory neurons have a little different shape, but this is their kind of dendrite area, so they detect stimuli out in the periphery. That information then is sent, this is part of the dendrite, into the nucleus, and then this is the axon itself, which will then make contact with the nervous system. What are some other ways to distinguish neurons based on their shape? There's three general kind. There is the multipolar, which we kind of viewed as that stereotypical motor neuron. There are cell shapes called bipolar cells, which look like they have this long dendrite and then an axon. Bipolar looking like they have two things. We'll see these play a big role in, say, for example, the eyes. And then there are these unipolar cells, which kind of seem to have one particular process. And those sensory neurons have that unipolar shape. So let's look a little bit further. If these are the individual components of a neuron, let's look at how one neuron makes contact with other neurons and cells. So the neuronal cell body and dendrites receive information across a synapse. Again, that was a term that was coined by a famous neuroscientist called Sherrington. We'll talk about him in another time. But that postulated thing that Cajal thought, the synapse, turned out to be the case. We've more recently, with the invention of the electron microscope, have been able to visualize that there is indeed a space between the axon terminals of the what we call the presynaptic neuron, and then these dendritic spines on the dendrites of the postsynaptic neuron. Notice the nomenclature that we're using here. If the synapse is our main area of interest, there is that which is before the synapse, the presynaptic neuron, the sending the information, and the postsynaptic neuron, which is here in orange, which is the receiver of the information. So information is transmitted from the presynaptic neuron to the postsynaptic neuron through these individual connections between axon terminals and dendritic branches on the postsynaptic neuron. Let's magnify the synapse and look in a little bit more detail about what's going on. So here's our presynaptic axon terminal and the dendritic spine, the postsynaptic cell. We can see there's a lot of things that are going on here, but I do want to point out a couple of things. So in the presynaptic membrane, this area here, what you can see are being released are all of these little green circles. These are representing chemical messengers, the neurotransmitters. You can see that they are bound in these vesicles, and then they spill out their contents into the synapse. Now, those neurotransmitters are going to adhere to certain proteins on that dendritic spine membrane called receptors, and they are on the postsynaptic membrane, the area in between the presynaptic and postsynaptic surface is called the synaptic cleft. So all of this together makes the synapse. But this is the functional unit of communication between one cell and the next within the nervous system. Let's magnify a little bit further. These membrane-bound sacs filled with neurotransmitter are called the synaptic vesicles. Now the vesicles contain that molecule, the neurotransmitters, and when they release their contents, they basically they fuse to the membrane itself through a process you may be familiar with called exocytosis, and just spill out their content into that synaptic cleft. Now, those neurotransmitters then will spread across the synapse and attach to specialized proteins on the postsynaptic membrane called receptors. Here they're kind of shown as little gateways. One way to think about the neurotransmitter is a key that can fit into and open up these postsynaptic receptors. Now, a lot goes on at the level of the synapse, and I want to show you that the connections are at these little protrusions called dendritic spines, and what's really unique about the dendritic spines is they're constantly shaping themselves and reshaping themselves. They undergo what we call plasticity. That is, they change their shape based on experience, based on usage. You can form new connections by growing new dendritic spines. There's a variety of different shapes, which I'm illustrating here. But if we were to look at an image of dendritic spines, this is a confocal time-lapse series of dendritic spines in a hippocampal slice. You can see that they're constantly changing just over time. Neuroplasticity refers to the continual remodeling of neuronal connections, and those connections occur at the level of the dendritic spines. We can look at dendritic spines through the microscope and determine their maturational state just based on their shape. These filopodia, these long, thin things, are very immature, whereas these are more mature. The other main cell type in the nervous system are the glial cells. Glial comes from the Latin meaning glue. They were originally thought to just be supportive cells holding kind of the major cells, the neurons, together and in their place. But it turns out glial cells have a number of important functions, and without them, the nervous system would cease to function. So what are these glial cells? I want to talk about them in general and then give you some specific examples of the different types of glia that are within the nervous system. So glial cells assist neuronal activity by providing raw materials, that is proteins or structural support, chemical signals, structure, and participate in information processing. One group of really important glial cells form an important component that acts as insulation around axons, and the insulation is called myelin. Myelin is kind of the plastic cord around you, or plastic covering around your electrical cords. It provides insulation necessary for electrical conduction, and it's made of a fatty tissue that's produced by different types of glia. Now, the two types of glial cells that produce myelin in the nervous system are, one, the oligodendrocytes, and two, the Schwann cells. Now, oligodendrocytes form myelin within the brain and the spinal cord, so they are specialized glial cells within the central nervous system. Schwann cells are another type of glial that produce myelin, but are in the peripheral nervous system. They're on the outside of the brain and the spinal cord. Now, how do you remember some of these names? Let's go ahead and show you this next image here. Now, an oligodendrocyte is just the single cell, and it has kind of these different arms that are processing out, and they wrap themselves around the axons of neurons, so that oligo means many, and dendro means branches. So the oligodendrocytes are cells with many branches that wrap themselves around axons, forming that myelin sheath. And you can see that there's some spaces in between one group of myelin sheath and the next, and this has a specialized name. It comes from that French physician we mentioned earlier, Ranvier. So these are the nodes of Ranvier. We'll see that this has a very specialized function in a later lecture. If we were to cut these axons with myelin down, we would see that the myelin is actually wrapped around and around and around the axon, forming kind of a layered structure. Okay. Schwann cells are just a single cell that wrap themselves around, so they don't have these branches. But again, the oligodendrocytes are in the central nervous system. The Schwann cells are in the peripheral nervous system. They're both producers of that myelin sheath around axons. Another type of glial cell are called the astrocytes, named because of their star shape. You can kind of see here. Now, astrocytes make contact with both capillaries, which provide a flow of oxygenated blood, and also deoxygenated outputs in the nervous system, and they make contact with neurons. So they stretch around and between neurons, and blood vessels secrete chemicals and help form the outer membrane around the brain. So that's one important type of glia. Another type are called microglia. Now, microglia are involved in the removal of debris from injured or dead cells. More recent discoveries in the neurosciences show that microglia and dysfunctions of microglia play a significant role in certain disorders like Alzheimer's disease. So if these specialized cells are damaged in a certain way, then they're unable to remove debris from the nervous system, and negative consequences occur. Okay, with all of that, let's go ahead and end this particular lecture with a retrieval practice question. So for this question, I want you to answer the blank are the input zones of neurons, and the blank are the output. So just name in order the input and the output of the neurons, and go ahead and indicate which answer it is. And we'll be speaking more about the rest of the nervous system in the following lectures.